Welcome back to the course Fundamentals of General Immunology, and today we'll discuss the remaining parts of molecular sensors of infection and cellular stress, and today is our lecture number 7. So just to recap, that we discussed that we have different kind of molecular sensors which can detect the pathogen associated molecular patterns or damage associated molecular details which are present on pathogens, and some of the cell associated structures like toll-like receptors, nod-like receptor, rig-like receptor, and C-type lactin-like receptors we discussed in the last class, and today we will discuss about the remaining receptors and also the soluble receptor, which are pentraxins, collectins, ficolins, complements, and natural antibodies. So we will start with scavenger receptors and we will move ahead. So scavenger receptors are again proteins which are present primarily on the macrophages and endothelial cells, and particularly involved in phagocytosis or endocytosis processes in order to detect the non-self targets. These are actually categorized into multiple types, and there are 12 types of these SRs reported. These are depends upon what kind of domains present on the protein surface. So for example, here we have this scavenger receptor. So it depends upon how many different kinds of receptor proteins are available. Say here we can say that this is a transmembrane domain, then this will be a cytoplasmic domain, and then this could be a recognition domain which can detect the pathogen associated patterns. And secondly, could be based on the cell type. For example, we could see that we might have the scavenger receptor present in macrophages and also endothelial cells. So let's try to understand this with an example here. So if you see, these are specific scavenger receptor present on a cell type, and these are actually working in collaboration with the toll-like receptors. So once they are identifying this specific molecules which are either present on pathogen associated molecules or could be a cellular damage events, then they can pass on this signal to this cytoplasmic protein called NF-kappa B. NF-kappa B is nuclear factor kappa B. It's a protein in the cytoplasm. This is always in bound form with inhibitor of kappa B. Now this inhibitor of kappa B and NF-kappa B is actually located in the cytoplasm. So to make this free, NF-kappa beta free, we need to remove this inhibitor from this NF-kappa B. And to inhibit this, we need to phosphorylate this inhibitor of kappa B. We have another protein available in the cytoplasm called inhibitor kappa beta kinase. So these are kinases which can do phosphorylation of inhibitor kappa B. So once this protein attaches to this inhibitor, it can do a phosphorylation. So essentially we see the phosphorylation happen here, and this become out, making this NF-kappa B freely move to the nucleus of a cell. And once this NF-kappa B goes to the cell, it's actually a transcription factor, and it starts doing transcription of different cytokines which can be involved in different inflammatory processes. So we could see that this inflammatory protein can do a lot of effect post its synthesis. So what we need to understand, that we have this inhibitor kappa beta kinase, and this inhibitor KB kinase can do phosphorylation of inhibitor kappa B, and this causes the NF kappa B to translocate into the nucleus and starts its function. So this is just a pathway representation of NF kappa B pathway, and the receptor is scavenger receptor. So we could look into this in details in our further classes when we encounter some of the things which are related to scavenging or scavenger receptor. All right, this one is a different receptor. It's called N-formyl methionine leucine phenylalanine receptors. This is actually a G protein coupled receptor. When we say G protein coupled receptor, it's a 7 transmembrane. So suppose this is a membrane, so this will be a transmembrane domain, and 1, 2, 3, 4, 5, 6 and the 7. So this is actually a 7 transmembrane domain that could be identifying some of the ligands from outside to the structure, and particularly these are identifying N-formylated peptides. N-formylated peptides are very specific peptides which are, or proteins which are produced by either bacteria or in host cell by mitochondria because of this endogenous, because of the origin of mitochondria. So just looking into the phenomena how this N-formyl peptides are produced. So we have this first amino acid that is coded by this codon AUG as methionine, and this methionine can bind to the formyl by this enzyme which are present called formyl transferase, and this enzyme in presence of a cofactor called 10-formyl tetrahydrofolate can convert this N-formyl methionine here, which can be the starting amino acid in a peptide chain coming out from a bacteria or mitochondrial species. So we have this peptide coming out from this bacteria can be identified as a foreign molecules, and these receptors which are G protein coupled receptor can identify that peptide as a foreign molecules on their surface. Sometime mitochondria also produces this peptide, and sometime if there is a leakage of mitochondria peptides, then this could be also identified as a foreign by the host cell to produce an immune response. So this is one such unique receptor that can identify N-formylated peptides. So we'll just try to look into how this is happening, some specificity. So we have this N-formyl methionine leucine phenylalanine receptor. So there is damage in the membrane of mitochondria or the bacterial species can efflux lot of N-formyl peptides into the system that goes with the blood circulation and goes to the different parts of the body, and in the blood circulation they may encounter neutrophils, and these neutrophils can have this G protein coupled receptor that can activate the downstream signaling pathway leading to activation of RAS pathway or calcium specific biology, which could lead to activation of other cellular processes. Now this phenomena could be highly synchronized and can activate neutrophils. This may cause inflammation, and this may cause kind of a deviation of the neutrophil biology based on what pathway it is activating. So this is a very complex biology, but for sake of study here, we just concentrate our understanding that these N-formyl peptides are produced by bacteria or by mitochondrial. So mitochondria is what producing these two species are only producing this N-formyl peptides, and these are detected by this receptor. So we'll try to wrap this cell specific receptor and move on to the soluble mediators which can sense some of the foreign molecules, and we'll first move to pentraxins. So as name itself indicates, these are pentameric structures, so these are cyclic pentamers, and this can react to the ligand in a calcium dependent fashion. So these are calcium dependent molecules or proteins. So pentraxins interact with the complement system and activate the immune system. We will see a little later that how this complement system works, what are the proteins involved in complement in a short while. There are two classifications of pentraxins. There are two classifications of pentraxins: short pentraxins and long pentraxins. Short pentraxins are produced in the liver, such as C-reactive proteins and serum amyloid P component SAP. So these C-reactive proteins and serum amyloid P component SAP is actually an indicator of liver specific inflammation or any inflammation that could upregulate the C-reactive proteins in a body can give an indication that there is something wrong with the body system. The second is long pentraxin. These are expressed locally by tissue system, either macrophages or neutrophils, and most common example is pentraxin 3 as a prototype of the long pentraxins. These are also involved in inflammation, and these are also critically expressed in multiple conditions. So, these are few examples like C-reactive proteins, serum amyloid P components, and pentraxins, other kinds which are related to soluble mediators of sensors. The next one is collectins or collectins. The collectins are specifically collagen containing C-type lectins. These are again soluble as the pentraxins are. These are also part of innate immune system. So, just remember that these are C-type lectins, soluble components, and very specifically they can recognize the carbohydrate structures actually. So, they can recognize the carbohydrate structure present on the microorganism to trigger its degradation either by activating the complement proteins or by phagocytosis. So, these are again very specific proteins which can detect the carbohydrate structure on a protein. There are few examples of it. The mannan binding lectins, these are produced by liver, and it actually activate the complement pathway, which we will see little later. And apart from that, we have the surfactant protein. The surfactant protein A and D, these are found in different areas of body, and few of them are lung alveolar fluid to give surfactant activity there, and to protect the body from the pulmonary infection, which regularly encounter multiple pathogens coming out from the respiratory system. The other collectins are the liver specific collectin called CLL1, and also the placenta specific called CLP1, and also kidney specific called CLK1. Again, these are all soluble mediators and can bind to the carbohydrate structures present on the pathogen. So, just giving an example that how these collectins look like structurally. So, we are just quoting here the example of surfactant protein A and D, and we could see that they have a carbohydrate recognition domain here in the top, and then we have a small nick domain there, and then there is a collagenous domain, and finally the cysteine rich domain in the bottom, and then generally they form a trimer, and then again more complex structure to make a functional unit of the surfactant protein. While if we see the D protein, actually this is a straight protein. Again, the contents are same. So, they have a carbohydrate recognition domain on the top, then they have a nick domain. Further down to that is a collagenous domain, and then the cysteine rich N-terminal domain. So, this is a monomeric structure, and then generally it is form a more complex trimeric structure, which again form a multimeric complex structure to start the complex phenomena. So, this is just the structural feature of collectins in a diagrammatic manner. The third one is the next one is ficolins. It again consists of collagen like region, but particularly the recognition domain is fibrinogen like domain. So, the C terminal is fibrinogen like domain. It recognizes pathogen through a very specific acetylation process. So, the acetylated molecules which are present on the pathogen, for example, N-acetyl glucosamine, they can detected by the fibrinogen like domain present on this ficolin, and it's activate the very specific MBL associated serine proteases, and therefore it starts doing the function. There are different kind of ficolin, there are different kind of ficolins present in the human body. M is present in leukocyte and serum, L ficolin which are present in serum, and it definitely binds to the acetylated components of a pathogen. And then finally the H ficolin, which found in again serum, and it's considered as the most abundant ficolin in the human body. So, just for the knowledge, three things to remember: that these can bind to acetylated molecules, and there are few of them which are present in human body, and the most abundant one is H ficolin, and these are abundant protein in the serum. So, this is one such soluble component. Finally, we would discuss a major protein which is called complement. So the complement is actually set forth of protein. Many proteins are there. These are present in plasma, and these are heavily glycosylated protein. The production site of this complement is mostly liver. So these are produced in liver. These are glycoprotein in nature, and their numbers are many. They circulate in blood as an inactive precursor, but will be activated by the production of a cascade that could start post entry of the pathogen. We could look into that phenomena, and this complement can do multiple function in order to clear the infection from the body. So this pathway of complement, actually we call it complement activation pathway, is actually there are three main pathways. All comes to the single point where there is an activation of this protein number 3 called complement protein 3, C3. Is the most abundant protein in the complement system. So the three pathways which are listed here is mostly based on how the C3 is getting activated. The first one is called classical pathway. These are triggered by antigen antibody complex. We have not gone through antibody, but we have understood the antigen. So the classical pathway is triggered by antigen antibody complex. The alternate pathway is initiated directly by the contact with the microbial surface component, and then immediately the C3 is getting hydrolyzed. And this lactin pathway is actually activated by MBL, which is a managed binding lectin present on the pathogen surface. So these are the three pathway: classical, alternate, and lectin specific pathway to activate the complement protein. So let's try to see what are the activation pathways and how it has been doing the phenomena in order to clear the infection. So these are the three pathway. The alternate pathway is actually, so the first step in alternate pathway is binding of the complement protein to microbial surface or the antibody. So here we have a microbial surface, and the C3 protein binds to this surface and getting cleaved out. This cleaved portion, so this is a C3A component, the smaller one, and the rest one is attached to the microbes, and then there is another component called B component that comes and bind to the C3 remaining fraction to make it C3 convertase, and finally the C3 is getting cleaved into C3b and C3a, which in turn make it complete C5 convertase, which can again cleave the C5 protein into C5b and C5a. So if you closely look into this pathway, so the ultimate aim is to make the C3 convertase and C5 convertase. So one C3 convertase, once the C3 convertase is made, the C3 convertase can cleave the C3 protein into active form C3b and C3a, and C3b in turns make a complex to form the C5 convertase. So this is basically a phenomena, and what we have understood in the previous slide, that the most important conversion to all this pathway is how the C3 convertase is made. It's a common phenomena. Classical pathways actually starts with antigen antibody complex, and we can look into this antigen antibody complex binds to, so this is actually an antibody which we will see in later classes, that can bind to the microbial surface. Antigens are present to microbial surface, they can bind and can activate some protein called C4 and then C2. They C4 and C2 again cleaved into its part like C4b and C4a, and C2 can cleave into C2a and C2b. C2a and C4ba can form and make this complex called C3 convertase, which again do a similar kind of cascade to make a C5 convertase ultimately. So C5b and C5a is ultimately produced in the classical pathway as well. The third one is lactin pathway, which is actually activated by mannan binding lectin, and they can do exactly the similar cascade which is reported for the classical pathway, but the only difference is the activation is happening here because of antigen antibody complex, and here it is because of the mannan binding lectin binding to the antigen present on the microbes. But then the activation pathway is same, that it can convert the C3 into C3 convertase, which ultimately leading to conversion of C5b and C5a. So, so ultimately all this pathway can lead to a degradation of the microbes, and how it happens we will see in the next slide. But for take home in this slide is just this molecule called C5b, that is got produced in all this pathway by C5 convertase. This C5b can go to a next round of this complement pathway to make this complex called membrane attack complex. Now, this membrane attack complex is a very important and last stage of complement pathway, where other proteins such as C6, C7 comes into picture, also C8 and C9. Collectively this C6, 7, 8 and 9 comes into the contact of C5b and make this specific channel on the microbial surface. And because of this channel on the microbial surface, there is a leakage of the content because there is an influx of the fluid happening inside the cell of microbes, and ultimately this can lead to the damage of the pathogenic cell. So, essentially the complement proteins are actually binding to the cell surface by doing multiple synchronized pathway to make a membrane attack complex, and once the complex is formed, the fluid can be say entered into the system to make this system rupture. So, the microbial cell can be destroyed by formation of membrane attack complex. So, just summarizing the whole complement pathway, that there are 3 pathways, and all this pathway converges to the point that how we could activate the C3 protein, because this is a common protein in all this pathway. The pathway can be start by a different mechanism, such as it could start by directly binding to the host cell. It could start with the binding to the microbial surface, or by antigen antibody complex, or by identifying some structures such as manase binding lectin on the surface of our microbes. But ultimately the purpose is to kill the pathogen in a very synchronized way. Now, look into what are the complement does in order to clear the infection. So, these are the 3 outcomes of complement activation. So, the first one is called opsonization. So, opsonization is a process where the complement protein can bind to the pathogen to make it phagocytized by a specific cell in the human body. So, here we could look into this process where the microbes can be bound with this protein C3b, and this can be recognized by the receptor of C3b present on the phagocyte, and once it is recognized, they can be engulfed by this phagocytic cell. So, essentially the binding of the complement can facilitate the killing of the pathogen by phagocytosis by the process called opsonization. So, this is one phenomena. The second is destruction of microbes by leukocyte, and how this happen is binding of again C3b to microbes release the a different kind of counterpart. When we discuss that when there is any protein of complement, it's cleaved into 2 fragments such as C3b and then C3a. Now, what is happening to C3a is actually anaphylatoxins. So, this can do multiple functions. So, C3a or C5a can recruit and activate the leukocyte by as a receptor biology. So, essentially the leukocytes are having a receptor for C3a and C5a. Once they are binding, the activation of the leukocyte can kill the pathogens or microbes. So, essentially in this pathway we can see that this minor component of the complement cleavage such as C3a and C5a can activate the leukocyte by the receptor mediated interaction, and once there is an interaction with the receptor, we could see that there is a destruction of the pathogen by specifically activating the leukocyte. And finally, there is either direct lysis of the bacteria by osmotic disturbances. So, essentially what we understood that the final step in the complement fixation is the membrane attack complex formation. So, once there is the complement protein coming to the microbial surface by all this pathway, alternate, classical and lectin pathway, can lead to production of these pores, and these pores can do an osmotic disturbances in the bacterial cell, leading to destruction of bacteria. So, these are the three basic outcome of complement activation. So, the basic thing which we need to remember is the complement is the protein present in serum and plasma in most abundant amount. These complement proteins are activated by multiple pathways, and all this pathway converges to a normal point where the C3 is getting activated, because this is the most abundant protein in the complement system. The activation cascade goes in a way that there could be a complement protein B and A, and this can be utilized in the further cascade. The alternate, classical and the lectin pathway can only differentiate it based on how the C3 can be activated. And finally, once we activate all this pathway, leading to a production of a membrane attack complex on the surface of our microbes, which can cause killing of the microbes in a very specific way, that could be because of osmotic lysis. The other two outcomes with complement is listed here, that either this phenomena of opsonization can happen, and in this phenomena the complement protein can bind to the microbes, and binding of the complement protein to the microbes can facilitate its phagocytosis by phagocytic cells such as dendritic cells, and because of this process the microbes can be cleared out from the human body. And otherwise, secondly, we could also see that the minor variant of the complement cleavage proteins such as C3a and C5a can bind to receptor to activate leukocyte, which can cause direct degradation of the leukocyte, which cause direct degradation of microbes through leukocyte biology. So, this is all about complement. And finally, we will come to this last class of soluble mediators, and that is called natural antibodies. So, when there is an infection, we could see that there is a generation of antibodies in the human body, and even though without infection, the body may contain different kinds of immunoglobulins, which we will see in the later classes. So, these immunoglobulins may be pre-existing immunoglobulins in the human body. Mostly these are IgM, but there could be an IgA and IgG immunoglobulins as well. So, we will see the classification of immunoglobulins in the later classes, but for time being, just to understand the natural antibodies are the antibodies which are present in the human body even without any infection, and these are produced by a specific subset of the B cell. And even there is no infection, they are present and circulating into the bloodstream to make the system sterile. So, this is actually a kind of host, this is kind of a mechanism which host cell does in order to clear the infection, even though without any sensitization of other immune system. So, these are the final soluble mediators of our senses. So, these are few of the examples of the molecular sensor present in the human body. Either they are cell associated or they are present in the soluble form. The whole idea of these sensors present, or whole purpose of these sensors in the cellular system is to clear the pathogen by binding specifically to their recognition molecules. This could be carbohydrate, this could be any kind of acetylated surface protein, this could be whole pathogen as such, there could be a soluble substances released from the pathogen, and we need to emphasize here the point that this not be only the pathogen, but also there could be a chance that there could be a possibility that the damage in the cell or the damage in the any tissue type can secrete certain substances, and these substances can also raise an immune response by binding to these specific cellular and soluble receptors. So we'll move on to the next discussion on our module 3, where we'll discuss about further classification and the details of the immune system. Thank you.